Thank you to the Society for making this presentation possible, and to the virtual audience attending today. I am presenting to you our findings on lipid compounds present in peanuts using the metabolomic approach. First, let me review the experimental design. The two major market types, Runner and Virginia, were obtained from commercial warehouses in Georgia and Virginia. For statistical relevance, samples were obtained from 3 different buying points for each market type, and 5 individual lots of 4.5 kilograms per lot each from each buying point were obtained. This produced a total of 30 samples. Each sample was analyzed in both the raw and the roasted state. The samples were subjected to a series of analyses, both targeted and non-targeted. Targeted is testing where the analyte is specifically analyzed for and the content is quantified. Non-targeted is the metabolomics approach, where the sample is subjected to a general non-optimized extraction, and then the extract is analyzed using both gas chromatography and high-pressure liquid chromatography with mass spectrophotometric detection. The principle is to identify as many compounds as possible without trying to quantify them, as the extraction may not be optimal for a particular compound. The data may be able to describe that a particular compound is increased or decreased by an effect, be it market type or processing, but the actual concentration is generally not reliable. This approach has been heavily used in the life sciences for bodily fluids, growth treatments, and so forth. For this study, the goal was to identify the compounds present in peanuts and to interpret how they differed between the market types. We can use the data to identify compounds to then target them for specific analytes. We can also trace them to their biochemical pathways. Hopefully, those working in marker-assisted breeding programs to assign genetic control to the compounds will find this data useful. Our focus will be for the compounds that are involved with flavor and flavor formation and nutritional content. From the study, 365 distinct compounds were identified. The compounds were assigned to their overall superpathway that would produce them. The highest number were from the amino acid and the lipid pathways. For this presentation, the lipids will be the concentration. Many of the compounds identified were based on fatty acids. The majority of the fatty acids in peanuts are palmitic, that is 16 carbons in length, and those with 18 carbons, in particular oleic and linoleic acids. Therefore, it was expected that the compound C would contain fatty acids of those lengths. The example structure presented here shows a fatty acid with 18 carbons in length with the normal positions of the double bonds. Oleic acid would have a double bond at the 9 position, and linoleic acid with double bonds at the 9 and the 12 carbon positions. The largest concentration of the lipid compounds are triglycerides, which is what is commonly identified as the fat content. In most cases, it constitutes 95% or more. Other compounds seen with the glycerol backbone were diglycerides containing 2 fatty acids and monoglycerides containing only 1. In almost all cases, the fatty acids present were either 16 or 18 carbons in length, especially palmitic and oleic or linoleic acids. Roasting would increase their content due to heat hydrolysis of the triglycerides, although they do occur naturally as precursors to certain phosphorylated compounds and biochemical signalers. The fatty acids themselves are degraded by oxidation during roasting to produce compounds such as aldehydes that have flavor impact. This leads to another group of compounds containing fatty acids found, that is the phospholipids. In these cases, the fatty acid is bound to a phosphate group that is a phosphorus atom with 4 oxygens. These compounds are usually found in membranes of cells and control membrane fluidity. Their nutritional properties include delivery of their associated fatty acids to cell membranes, where they can be used for lipid synthesis or as cell messengers. These compounds are often used as emulsifiers in processed foods because of their solubility in water and oil blends. A related type of compound are those where the glycerol backbone has been replaced by amino acids. These are the sphingolipids. Soybeans are known for these compounds, so it was expected to see them in peanuts as well. Like those based on glycerol, these compounds are membrane compounds involved in signal transduction. They are induced by abiotic stress during seed formation. Nutritionally, those from soybean have been found to be anti-inflammatory. These compounds were also found to survive the roasting process in our study. Hydroxy fatty acids are functional oxylipids. The ones found were the 9-hydroxy octadecadienoic acid and 13-hydroxy octadecadienoic acid, which are normally seen together, as well as in the 9,10 and 12,13 dihydroxy octadecanoic acid pair. These are seen in the raw as well as the roasted and could possibly be induced by abiotic stress. In animals, these compounds are induced by cold and stress on muscles. They induce the uptake of fatty acids by cells to repair tissues. They are more reactive, have greater stability and viscosity, and mix readily with various solvents, more so than fatty acids, and so they have industrial applications. Fatty acids bound to amine groups were also found. These compounds go further to convert the peanut fatty acids so that they will be found in the growing seed. In this study, they were found at higher levels in the roasted samples, so they also can be created by heat polymerization. Short-chain fatty acids are usually breakdown products in the roasted samples. They are often the result of oxidation of the long-chain fatty acids at the site of the double bond. They can also result from bacterial attack. They are associated with off flavors that are described as rancid or stale. Phytosterols are the plant equivalent of cholesterol in animal lipids. They differ from each other in the structure of the side chain at the position designated in the figure on the slide by the letter A. Beta-sitosterol is the one present in the largest concentration and is the one pictured here. In cells, they provide rigidity to the membrane, control movement of components such as enzymes in and out of the cells. Nutritionally, they are advantageous in that their similarity to cholesterol allows them to block absorption of cholesterol from the intestine, either due to blocking the absorption site or by taking the place of cholesterol. Beta-sitosterol has been found in the blood of subjects after consuming foods containing plant oils and has been associated with reduction in inflammation. Phytoalexins is a term which covers a range of compounds. The ones identified by this study were compounds such as resveratrol and related stilbenes. They are created via the lipid pathways as a reaction to some sort of stress, such as fungal attack. They are of interest as there have been many studies of their health effects. Their presence is usually environmental rather than genetically related. Tocopherols, or vitamin E, are one of the most important fat-soluble nutrients present in peanuts. In many cases, tocopherols are complex to fatty acids as they provide antioxidant protection to the double bonds. Alpha and gamma are found in close to equivalent amounts in peanuts, usually at 100 to 200 micrograms per gram of oil. Alpha is the form that is nutritionally available to humans. The gamma form provides more antioxidant protection for the peanut itself. The beta and delta forms are present at much more lower levels. In this study, the amounts were actually quantified using a targeted analysis. The information found is important in that the total levels of tocopherols were lower in the Virginias when compared to the runners. When this study was set up, normal oleic runner peanuts were requested from the suppliers, but in reality, some lots were normal and some were high oleic. From the tocopherol data, the cultivars actually clustered according to their type when using principal component analysis. This shows a relationship between fatty acid content and total tocopherol content. The high oleic lots also had higher gamma tocopherol content than alpha tocopherol. This explains the relationship of fatty acid composition to tocopherol content. To summarize this discussion, there are many types of lipids present in peanuts other than triglycerides. They are involved in all types of biochemical interactions in peanut metabolism. Of greatest interest to our research are those that contribute to flavor, either as precursors or processing results. The small-chain fatty acids are responsible for sour and acidic flavors that are usually only seen with extreme degradation. Large intact fatty acids do not contribute flavor, but rather more to texture and to mouthfeel. The oxidized fatty acids produce rancid or painty, oily, and stale or cardboard flavors. From our study, we learned a great deal about peanut composition, but probably the genetic control of lipid composition would not be a greater contributor to roasted peanut flavor. I would like to thank Birdsong Peanut and the Golden Peanut and Tree Nut Company for supplying the peanuts for this study. The Buehler Aeroglide Company provided roasting equipment for the sample preparation, and Metabolon participated in the non-targeted analyses and the data interpretation. Thank you for your attention to my presentation. Please contact me by email with your questions and comments.